Welcome to the final night of Pint of Science in Armidale. The first time that this national science communication and wonderful night of beer and science together has been run in Armidale. And we're sharing the night with many other regional and capital cities around Australia. So hello everyone around Australia. And in fact, you guys can help us say hello. We're videoing tonight. We videoed last 2 nights as well. And Pint of Science Australia want to hear from you too. So I think, Ian, are we going to say hello from everybody? Can you go around the room? Can I get a big hello from everybody? After 3, can everybody just say hello from Armidale, Pint of Science? Yeah? Do you want to practice? No, we can do it. Okay, so after 3, hello from Armidale, Pint of Science. Ready? 1, and look at the camera if you can, sorry. 1, 2, 3. Hello from Armidale, Pint of Science. Thank you. Great start to the evening. So, do you change the slide here or do I, Ian? You will. Great. So welcome to the world in the Wicklow tonight. We've had 2 fantastic nights of science. Monday being, it was a chemistry night, and I saw the main talk, Dr. Liz New from Sydney Uni, and she was amazing. She did this fantastic contextual talk about fluorescence in nature and then her cancer therapy drugs. It was awesome. Last night, I wasn't here, but I hear it was packed just like this and awesome. Sorry, I pressed it and I shouldn't have. Great. And last night was Guts, In Your Guts, Microbes, and the 2 speakers. We had Gull Winter and Dr. Rob Hart, and they were amazing as well. And tonight we're finishing off with even more amazing talks, and we're sort of in an environmental science focus tonight. So thank you for coming and showing your support for that sort of science in Armidale. And I knew it was going to be a good turnout, because it will be for the rest of the year too. My name's Kirsty Abbott. I'm an ecologist, but work with education at UNE and lots of science communication too. You might have seen me at Science in the Club, and we'll be doing Science in the Club this year, which I'll talk about too. But for tonight, I'm your MC. I'm going to be helping you through trivia questions. I'm going to be asking trivia questions. I can't answer them for you. And leading you through some Q&A at the end as well. So turn your trivia brains on now so that hopefully by the time the questions come, you'll be in good trivia mode. To start off though, I want to thank all the organisers. Pint of Science is a huge event around Australia, and it couldn't happen without the support of CSIRO. From Inspiring Australia, the New England Northwest Regional Science Hub, from Ecological, our sponsors tonight, from University of New England, the School of Science and Technology, and of course the Wicklow, providing a new venue for these sorts of events in Armidale, which we'll continue using this year, which is great. So thank you very much. Also, before starting this evening, we would like to acknowledge the traditional custodians of the land on which we gather and pay our respects to elders, past and present. We hold great value and respect for Indigenous knowledge systems as a vital part of the knowledge capital of Australia. And in fact, the environmental science focus today, I think, is really lovely to reflect on the fact that Indigenous Australians were the first stewards of the land, and in a way, we're really just catching up. All right, so without further ado, I am going to introduce our first rapid-fire speaker. First of 4, Sajani, please come up and tell us a little bit about soil pollution and kidney failure. Please put your hands together for Sajani.
Okay. Will the crisis of soil pollution turn Sri Lanka into an island of widows? Farming community in Sri Lanka is now in a crisis. What is this crisis? An extraordinary outbreak of kidney failure leading to death. The disease has largely been confined to the male farmers. What may be the reason behind this? Well, Sri Lankan farmers have been applying heavily contaminated phosphate fertilizers since 1967. The major contaminant identified is cadmium. Cadmium, very toxic heavy metal. In addition to that, other agricultural inputs or other agrochemicals such as insecticide, herbicide, contribute soil pollution with arsenic. Arsenic, a very toxic contaminant. The most interesting thing is the behavior of arsenic and cadmium in the soil system. It is totally different. Okay. So, yes. Studies conducted by World Health Organization and scientists in Sri Lanka have isolated soil cadmium as the key cause for this problem. Arsenic also has been identified as a contributory factor. Unfortunately, we don't have any solution for this problem yet. That is the purpose of my PhD, to find a solution for this crucial problem in Sri Lanka. Based on the information available, I predicted that the effective remediation of farm soils in the affected areas will be a solution. So, I am going to use effective natural sorbents to neutralize this contaminant. Biochar, a kind of charcoal, soil organic amendment, such as compost, will be the sorbents which are going to be used. In this study, I'm going to investigate the changes in behavior of arsenic and cadmium in the soil ecosystem with the application of different biochars and different organic amendments. Biochar and soil organic amendment will be a cost-effective solution for the soils contaminated with these toxic metals. Finally, I hope that this cost-effective solution will save thousands of lives of people in the world. Actually, more than a hope, this is what I believe. Thank you.
Thank you so much, Sajani. Please write down any questions you have about the cadmium-arsenic interaction now so you don't forget for Q&A later. But that's fantastic to start off with such a massive intention to save people's lives through this. It's fantastic. All right, moving on to rapid fire number 2. We have Katie Pine, The Hunger Games. She's going to tell us about catching carbon. Please put your hands together for Katie.
Okay, so before I start, I just want a show of hands. Has anyone read or seen the Hunger Games movies? Okay, so half of you will know what I'm talking about tonight. Excellent. The rest of you will see how we go. Can't promise anything. Okay, so we live in a carbon-hungry world. Most living organisms are made of carbon, and a lot of man-made products contain carbon, most notably fuel. Okay, but one of the things about carbon is that it's not renewable. Yes, it does exist in different forms, but ultimately, once we've used it and it goes into the atmosphere, it's really hard to get back down to Earth. In terms of the winners of carbon, at this stage, it's looking like the atmosphere is the winner. Okay, so they're like the capital in our Hunger Games scenario. Okay, they've got all the carbon, they're calling the big shots, most namely climate change. Okay, so we've got increasing temperatures and so on. The losers at the moment is the soil. So soil organic carbon, especially in Australia, is really low. This is our District 12 of the Hunger Games, okay? So they got nothing. They're trying to make do with what little they have. All right, so this is putting increased pressure on farmers to be able to produce more food and fibre to feed a growing global population, but at the same time trying to make sure that there's carbon staying in the ground and that they're not losing any other nutrients. Okay, so it's a pretty tough call. So some of the things that industry is doing, in particular I work with cotton, and we've been looking at introducing rotational crops. So at the study site I'm with, for the last 20 years or so, they've been trying to rotate cotton with maize, okay, so it's a carbon-rich source. And basically, they're hoping that they get a return or carbon stored about 20 tonnes per hectare. And that's the whole aim. So every second year, the maize crop goes in instead of the carbon. introducing, hopefully, carbon into the soil. Unfortunately, some of the work we've been doing has been showing that that's not the case. Yui in the audience here, if there's any carbon questions, she'll address them for you. And so we're wondering, where is this carbon going? What's happening to it? And some of the work I've been doing, I'm coming to realisation that soil microbes could actually be a key player in this. They're a bit of our unknown, or our District 13 in the Hunger Games. You can't not quite know if they're the goodies, the baddies, what their motives are. This is the soil microbes. So soil microbes are really important in soil systems because they help transform vital nutrients for plants, especially in the cotton plants, so transforming nitrogen and phosphorus for them to use. Unfortunately, for them to do this, they need an energy source, and guess what that source is? Carbon. Okay, so like you or I, when we consume carbon and other energy, we exhale carbon dioxide. Same thing happens with the soil microorganisms. So part of my work now is to try and figure out if the soil microbes using this carbon and turning over these nutrients is actually beneficial to the system. So are the soil nutrients more important than the potential carbon lost into the system? So who's going to win this? Is it going to be the carbon-hungry atmosphere, our capital, or the poor District 12 soil carbon, or our unknown players, our District 13 soil microorganisms? That remains to be seen, but may the odds be ever in their favour. Thank you.
Thank you so much, Katie. I love that the first two talks on this are sort of from the ground up and about soil health, and you know, it's so important for everything else we do on top of the soil, right? It's great. All right. Again, write down any questions you've got now so they don't leave your head for later. And I'd like to introduce our third speaker. Talisa's going to talk about remote temperature monitoring in sheep. Please put your hand together for Talisa.
Thank you. Hello, everyone. I'm working on a project funded by the Sheep CRC, and we're looking at ways to remotely monitor health of sheep in extensive environments. So if you've got a sheep farmer, he loves his sheep, not too much, just platonically, but, you know, he likes his sheep, he wants them to be healthy and happy and everything. and so if you look out in the paddock, you don't know if sheep number 53 Fred over there is getting crook and, you know, he's sneezing on everybody else, making everyone sick. You don't know. They look quite happy at the moment. So we're looking at ways to actually work out how healthy they are and how well they are during while they're still in the paddock, so that we don't have to chase them around and grab hold of them and talk to them, and they don't really want to know us. They just want to go and eat grass. So one of the things that I'm looking at, one of the normal health monitoring aspects when you take your animal to the vet, the first thing they do is stick a probe where the sun don't shine and get a temperature. So we want to actually do that without having to chase down each sheep and put a probe there. So I'm looking at technology and devices which are able to monitor body temperature out in the field. It's nerd paradise pretty much. I get to play with animals and technology and gadgets. So it's lots of fun. So one of the aspects that is part of my project that I'm looking at is temperature sensing microchips. They're the same as the microchips you put in your dog or cat, but they actually have a temperature sensor as well. So we're looking at ways of placing those in the sheep so that when they come to a reading point, we can actually get a temperature for each day, and we're analysing whether we can actually use that to determine whether they are healthy or whether they're starting to get sick, and so that a farmer can go and monitor them straight away. And another aspect of technology that we're actually using is infrared thermography, which is a lot of fun, and it can actually determine whether there's a surface temperature issue. So one of the things that we want to find out is whether we can actually determine whether a sheep is coming down with fly strike, for example, and that's a surface inflammation caused by flies laying their eggs on the skin, and the maggots start to eat and do all sorts of horrible things to our poor little sheep. So we want to be able to pick that up before too much of him gets eaten. And so this is what we're actually looking at. So I've got a couple of images there. One with a control sheep, normal sheep, nothing wrong with him. And then on the other side, we've got a little bit of fly stroke starting up. So one of the issues, of course, with that is trying to figure out whether we can actually detect any temperature changes underneath the wool. It turns out wool's really insulating, so maybe not so much. But we still would like to know if we can actually apply this technology in this capacity. And those are the two projects that I'm currently writing up at the moment. And it's some very, very interesting work. And I'm very grateful to the Sheep CRC and UNE for actually allowing me to do this project. So thank you very much for listening.
Thanks, Talissa. I hope you didn't hurt your knee running after sheep with a probe or anything, did you, your ankle? Good. All right, and I'm really proud to introduce our last speaker, Dane, telling us about plants and the quest for antibiotics. Now, Dane's done so much for Pint of Science. I don't even know how he's got it capacity to actually do a rapid fire talk already. So I'm in awe, Dane. Welcome. Please put your hand together for Dane.
I think Kirsty might have spoke too soon. I haven't actually given it yet. I could just run off right now, but... basically I'm really interested in microbes, so I'm a microbiologist in my original studies, and I love plants. I'm an avid gardener, so I had to combine my two loves and try and do something with it. So I went, well, there's a big problem at the moment, and that is superbugs. That's a bit of MRSA right there, which some of you have heard of, one of those golden staffs that just does really nasty things. And I was working in a clinical micro lab for a while, and in the old days, when we saw MRSA, this superbug, come into the lab, it was alert stations, everyone panicked. And as time's gone on, it's actually become almost just, another one. Yep, move on, another one. So it's becoming quite a big concern. And there aren't many antibiotics anymore that we can use to treat a lot of our bugs. So you might have heard, I think it was last year, a lady in the US died from a bug that was basically untreatable. She ended up, I think, started off with a urinary tract infection, something like that. And before you know it, things progressed very rapidly and they couldn't treat it. So basically there are a lot of people who need antibiotics but don't have any that are going to work because of this resistance problem. And Big Pharma has done, we can't diss them too much because they do a lot of research in this area, and it's a really hard area, but it's expensive, it's time consuming, and by the time you get your drug to market, you've already got resistance. So it's not really going to make you any money. So it's a bit of an issue. So that leaves a big gap in the research. And so basically researchers are having to step in and try and find new drugs. And the old methods was basically screening bacteria, try and kill other bacteria, and fungus kills other bacteria. So we used to steal their chemicals and use those as antibiotics. We've kind of hit the wall with that. So we're looking for new ways now. So we're a little bit desperate, but we're trying different things. So that's meant to be a leaf I tried to draw there. I'm looking at plants to kind of eye off these bacteria and try and kill them off. And that's clear by the BAM. I'm not finished my PhD and we haven't quite got a BAM yet, but we're going to get one, I hope. I can feel it. So that's the plan on my part. So I'm hoping basically to use plants, and plants are there's so many chemicals in plants, and they've had to evolve fighting bacteria just like everything else on the planet. So they're producing a bunch of chemicals. They don't have nice immune systems like we have. They rely heavily on basically chemicals, small chemicals, to kill their bacteria. So I'm hopeful that there's one or two in there that we can find that might actually do something good. The bigger problem is plants aren't humans, they're not animals, so a lot of the chemicals they produce will kill us too or make us very sick. And that's the biggest problem. So it's a bit of a challenge for us, but hopefully I'll get something that won't kill you all. The other problem is plants produce a lot of different chemicals and none of them are particularly strong. So there's a lot of chemicals sharing the work basically to kill bacteria. So I'm just hopeful there's just so many of them out there that there's got to be one out there. I'm optimistic, perhaps not my PhD, I won't get the one, but I feel that there's one out there. So basically what I do really quickly is I go out into the field and set off all of people's camera traps and things out in the... I'm sure I pop up in a few people's cameras out in the bush. Grab some plants and I do some extracts of those plants, so different ways to try and get out some of the secondary metabolites, so the chemicals that might be useful for me. And I just quickly test them and see what plants kill bacteria, what don't. And you can see here where you get a nice clearing. That's where I get really excited and go, oh, there's something in this that kills bacteria. Unfortunately, that's my antibiotic control, so big surprise. But something like this one that still does have a bit of a halo gets me excited. So the problem then is that my extracts are a huge complex mix of compounds, so I need to kind of sort that out. So I run this cool little plate, which is called a TLC, and basically you drop some chemical on it, or mix of chemicals in my case, and you still in some solvent. And if any of you have ever put a bit of pen on the bottom of paper and sat it in water and the water moves up and all the colours separate out of the ink, same kind of process. All my compounds separate out then. And then I grow some bacteria. I put some agar straight on the top of that card with some bacteria in it, and then I add a dye that changes colour if the bacteria is growing. So in this case, lots of growth here where it's kind of coloured, so I'm not interested in anything there, but there's this big thing going on here, which turns out to be cerilitane or something, I don't know. I'm not actually a chemist, so I end up doing all this chemistry stuff and having to know all my chemistry. So that's a lot of fun. But anyway, it's a cerilitane, and it's from a bush that's called emu bush. Well, it's not actually the normal thing we'd call emu bush, but it's from a family of plants called emu bushes, which is kind of cool. But that's still not enough. I've then got to separate those chemicals out en masse. So I do some things like columns and some fancy chemistry that I don't know I'm doing half the time and break things all the time. Then I get some stuff out, which is cool, and I've got a few things already, not a huge amount yet, still got a bit of time left. And then I can pop them in this big, really expensive machine the uni has that helps me identify what the compound is. And we can work out, for example, there's a compound there that's actually that really active thing there, I think, from memory. That's it there. And then after that, I need to know how active it is. So I use this plates at the end where I put basically a dilution along the plates. And where I get some colour, bacteria is growing, and where there's nothing, I've killed the bacteria, and I can see how active basically it is at what concentrations. And that's where I'm hoping to get to. And then, of course, we've got to do toxicity tests after that in cells to make sure we're not going to kill you. But that's it from me. Thanks.
Thank you. Thank you so much, Dane. And that brings us to the end of the rapid fire talks for tonight. Thank you, speakers. So, audience, please grab a pen, grab your voting sheet, and we're going to turn on the elevator music. Give some mood while you all vote. Please just vote for one talk. And are you going to go around and collect? And in about 2 minutes, Braden is going to walk around and collect your votes. All right, we have Q&A. I don't really want to start off the Q&A. I want questions to come from the audience. Would anyone like to start off with the first question for Guy or any of the rapid fire students? And Michael's got a microphone that he'll bring around.
Suze, I've got a question for Guy. I'm just wondering why the collars are set to fall off. Don't you want to keep tracking where they move? Or?
Great question. The collars fall off the dingoes simply because of battery technology. The batteries have not been up to long-term deployments until recently. Now we've added solar cells to trickle charge. We are reaching a point where in the next couple of years we'll be able to think of lifetime monitoring. We've then also got to think of the ethics of lifetime monitoring. But if we can make them small enough and they don't impact on the welfare of the animal, we'll probably move in the next few years to at least 3 or 4 year deployments.
Good one. All right, next question. And that's quite... So when you say lifetime monitoring, actually I'm just going to follow it. Lifetime of how long?
10 years.
Yeah, okay. Wow, that's significantly longer than now. Oh, no, so yeah. All right, we have another question. Anywhere? Yeah, at the back there. Oh, sorry, this one first, sorry.
Sorry, mine's for bacteria antibiotics. Sorry, I've forgotten your name. So we were just wondering about different types of plants. Are you just focusing on terrestrial plants, or are you using like sort of phytoplankton to look at things, or aquatic plants. As we know, like bacteria love warm, moist environments. So, and is anyone, as a side note, is anyone researching things like bacteriophages to sort of see if they can synthesize antibiotics in that way?
Yeah, so okay, there's a few questions in there, I guess. The first part of that is I'm looking mostly at local, mostly native plants from the New England region, but I'm also looking at some plants from kind of on the border of the desert, so aeromophilas, which are the emu bushes. They had a lot of traditional use for plant medicine, so there's some, we have a bit of an expectation that we'll find compounds in those, so that's why we choose those. More recently, I've started thinking more about plants that grow near river edges and things like that for that very reason, that that's a nice environment for bacteria as well. So it is something that's occurred to me, but I haven't actually really done much on that at this point. There are lots of groups doing similar research with especially water plants, not just water plants, organisms that aren't plants at all, but things like corals and things like that as well. And bacteria in marine sediments and things like that are giving a bit of hope. With bacteriophages, so bacteriophages have been used and there's been clinical trials, I think, around using bacteriophages. So for anyone who doesn't know, a bacteriophage is a virus that will kill bacteria, basically. It infects bacteria. There's a few problems with it. So you can't just use one type of bacteriophage, because the big problem is that when you go to your doctor, the doctor has a bit of an idea what you might have. They don't know exactly what organism it is. And even if they send it to the lab, if you're lucky, the lab will get it down to species level, the bacteria, but maybe they won't. And even then that takes time. And by then the patient's gone and it's probably not going to come back to the doctor. So antibiotics have the advantage that they usually have a broader spectrum, so they can kill a few bugs. With bacteriophages, we'd need to have a really rapid diagnostic test that we could know exactly what bug it was making the person sick, so that we give them the right bacteriophage for that. So there was a clinical trial I heard about recently where they're actually mixing a group of different bacteriophages to get around that. And there's some promise in that as well, definitely. So that might be the future, yeah.
Cool, thank you.
Yeah, I've just got a question for the person who spoke about the prevalence of kidney disease in Sri Lanka. Oh, I don't need that. Yeah. All right. I was just wondering if you could tell us, and I'm not sure, you may have actually mentioned it, but where's the worst of the cases in Sri Lanka with the kidney disease? Is it closer to the bigger population areas?
Yeah, in Sri Lanka there are three major places based on the climatic region: dry zone, wet zone, and intermediate zone. This is the place we call dry zone, because dry zone is the most popular farming area in Sri Lanka, especially paddy farming. Because the farmers, they use excess amount of fertilizers, especially phosphate and other fertilizers, as well as agrochemicals such as insecticide, herbicide, like agrochemicals. They use these things excessively because they think this is really important to increase the yield. So with that, this fertilizer and agrochemicals are polluted with cadmium and arsenic. That is the thing we identified, not me, actually, by scientists. So I'm going to find some solution for this problem, because already identified cadmium and arsenic are the major problem for kidney disease in Sri Lanka. Yeah, that is because of the overuse of these chemicals, agrochemicals and fertilizer for the farming, especially for paddy farming, because rice is the staple food, major food in Sri Lanka. So yeah, they use excess, and also when we consider the rice, the usage of rice, it's very high compared with other countries, because it is the staple food in there. So scientists believe that normally this cadmium and arsenic come from the rice as a dietary source, as well as through the water, and there are so many sources. Yes.
Are you aware if there are similar problems in the south of India?
Sorry?
Are you aware if there are similar problems in the south of India?
Yeah, yes. Yeah, as well as Bangladesh. it is arsenic. That's a problem. It's arsenic, and also India, as you mentioned. Yeah. But in here, the problem is this is not only from arsenic. This is from arsenic and cadmium. It's a totally different. This cadmium is a metal. Arsenic is not metal. It's a metalloid. And the behavior of arsenic and cadmium in the soil is totally different. That's a problem. It's very difficult to go for the solution with this. Yes.
Thank you.
Sounds like you've got solutions for more than just Sri Lanka. I think, Sanjani, or you will have. Any next question? Yep. And then down the front. Tamara.
What, Guy, what do you classify as a pure dingo? Is there certain traits you look for, genetics, something like that?
To be blunt, we rely on the advice of our geneticists. So there are accepted tests, and I mentioned before that there are categories 1 to 6 based on tests that look for association with a group of known genetic features. 90% or above considered pure, 10% or below considered a pure domestic dog, basically. So if one of the groups we use is Zool Genetics in Western Australia, and that's Danielle, who was one of our PhD students previously. If Danielle says, yep, it's come back and it's category 1 or 2, we label it dingo. But in the field, we don't try.
Actually, Guy, I'm going to comment on that too, only because I saw a talk. I saw a talk by this guy, Laurie Corbett. He like Mr. Dingo from a long time ago. And I remember him telling us that the only way you could tell a pure dingo was to kill it and scalp it and do a whole heap of morphological measurements on the skull. Is that? No, right. So that was my totally like baseline. That's the only way you can tell dingoes. And yeah, and then I saw all the skull people over here and wanted to know.
Oh, it's fun though.
Sorry, it doesn't work, but it's fun. Okay, cool. So genetic markers are the way to go. Right. Interesting. Okay. Question from the floor, down the front here. Yeah, from the floor.
Hi, I've got a question about the cotton and the maize. So microbes respire CO2, but some of them store carbon, like I'm thinking mycorrhizal fungi, they use carbon to make their mycelium, and that actually stays in the ground. Maybe that gets eaten later. But do you find, are there differences in how much gets respired, how much gets stored, and can you quantify that at all?
Thank you. So we're still in the early stages of this. This is something that hasn't been explored too much in depth at the moment. Basically, what I've been looking at is the activity of microbes down the soil profile, so from 0 to 100 centimetres, and looking at their activity as they go down. And what we've been finding is that when they've been growing under the maize, they are respiring more, so they're more active than when they're under cotton. So it's suggesting that the carbon from the maize is stimulating their activity, but we haven't yet defined whether or not it's certain groups that are doing it. You haven't identified species or genus? I have just got a ton of data back from metagenomics, which looks like something my toddler painted in preschool. It just came back with this blur of lines, and I'm currently sifting through it and having a look. So a lot of different bacterial species in there, a lot of diversity in that. Fungi, not so much. And there's only a few archaeal species in there. So hopefully in another year I'll be able to fill you in more.
We'll just schedule you in for next year, Pint of Science, to update us, shall we? Thanks.
Oh yeah, that sounds great.
Thanks, Katie. Another question from the floor somewhere, from the audience? Yep, down the front here.
Sorry, my question is to Guy. Guy, with the crossover with domestic dogs like dingoes, as Canis familiaris, other true species in the world of the canid species, do they have a tendency to travel as much as like the wolves that they're introduced in North America or reintroduced?
Yes. And wolves in particular have been recorded undertaking huge forays and dispersals. We've started offering some live tracking data to people in the last couple of years, and if anybody wants to go to Riverina Wild Dog Project, they can see 29 wild dogs data as they move around the landscape, have been for the last 12 months. Americans have been doing that with wolves, but over much bigger distances as wolves recolonise areas. And this is one of the questions. They're moving from source populations into what are areas that are essentially, in some cases, essentially free of conspecifics. So that might lead them further and further. But in Europe, there have been big dispersals of wolves too. Foxes, interestingly enough, and I know we spoke about foxes the other day, we've recorded foxes in the local area moving 25 and 30 kilometres. So they're a much smaller animal and they will still undertake large movements, and we've now seen it in cats too and in quolls. And as we collar more species, we're seeing these dispersals in a wide range of species, but again only often in a small proportion of the population.
Do you have a large population of wild dogs on your place, Richard? Yeah. Okay. Questions?
Stephen?
Yep.
On the dingo wild dog thing. Sorry, I missed. Sorry, I really enjoyed what I saw, but unfortunately I missed a lot of it. I'm really interested in these sort of knock-on effects with respect to taking dingoes and wild dogs out. And you may well have covered it, but to me, if you're killing them, there are going to be knock-on effects. The only question is, what are they? So sort of a first, just was interested in how you were analysing that or trying to get some sort of an idea of what's happening.
Sorry. Oh, now you've got one. Okay. Sorry.
We're going to have to try.
Okay.
So, yes, you did miss a little bit that's relevant to that. Basically, we have a replicated design that uses multiple study sites in the Northern Tablelands to look at different treatments. So we have some sites which have no control of dingoes and others where there's intensive control. So we've been able to look at what happens when densities of dingoes change, what happens to foxes, cats, medium-sized prey, herbivores, and we're looking at vegetation responses as well. In terms of foxes and cats, when dogs are removed using local practices, we see a decline in foxes at the same time. In fact, a disproportionate decline in foxes.
Sorry, to what degree is that because foxes are taking the same bait?
Yes. Yeah, so it's an immediate response. So it's an immediate direct response of control.
But of course, dingoes kill foxes too.
Not many. They just can't access them. And that's the sort of the sympatry that we've observed through the collaring that we've done. Foxes and dogs using the same environments, maternal dens less than 1 kilometre apart, overlapping each other in space but not in time. So there's almost certainly temporal partitioning locally, and that could well be a function of the extra structure that we have here, which affords protection for smaller predators from dogs, as opposed to the desert. We're not seeing any strong evidence. The stuff we've done has shown no significant relationship in activity between foxes and dogs or cats and dogs. And now we're going the next step using individual identification to see whether it holds out for densities as well.
Another question for Guy. Do dingoes in this area cruise around in packs? And if they do, are they like, is a pack a single family structure? And if that's the case, why don't foxes cruise around in packs?
We say social groups. Yes, dogs, northeast New South Wales, it's common for them to occur in social groups, but the fundamental social unit of the dog population is a pair. So they're just trying to form pairs. And then depending on the circumstances, that pair will have young with it. It may have one or two of last year's young and this year's young, and then some of them will disperse. But the fundamental is a pair, but we do see aggregations of up to 10, 12, sometimes 14, but that's almost always when neighbouring related groups come together, and usually they're sisters as mothers, and they're happy to sort of cache their kids together, and they'll wander around and socialise and then go their separate ways. In terms of foxes, again, we had some field days last week where I showed some data and showed some photos of 3 foxes wandering around together, and you sometimes get 4 foxes together. But again, that's almost always mum, dad and the kids. It doesn't persist long term, but it's not just while they're lactating and not just while they're feeding. It moves into a phase where there is teaching and some, I won't say cooperative hunting, but certainly there are a group of animals involved in feeding at the same time, and then they disperse.
Question up the back? Yes, you may. Try yelling, there's a microphone coming.
Hello. First of all, this is for Guy. I'm extremely interested in your area of research, and at the moment we're doing a unit on animal welfare, and you briefly mentioned the trap, and I put my hand in it too. It's fine. But I was wondering how you get through the ethics committee, how you work that with the boards as well, and like having to hold them down and stuff like that. How you manage to get them to let you through?
No, great question. And like anybody else, and I'm not being flippant, we answer their questions and provide evidence for what we do. And to be honest, we're probably very fortunate that our techniques look better than the old techniques. So Laurie Corbett and Alan Newsom used to use basically modified bear traps to catch. They send their techos out to catch dogs. They'd shoot them in the neck. So sometimes they'd die immediately and sometimes they wouldn't. And then they'd cut them up. And if we turned up with that as our proposal now, we'd struggle. And we don't. We turn up honestly saying, we have a reason for doing this work. We will treat these animals with care. They're important study animals to us. We care about their welfare. We've modified procedures iteratively as we've gone to make things better and better and better for the animals. I didn't say we rub a special ointment onto their foot to try and promote blood flow so they don't get ischemia, which can result in rotting in released canids, which some southern Queensland projects have demonstrated very well. We learn from those mistakes that other people have made by sharing information within the research community, and we're honest with the committee. If we have problems, we did once have a dog that damaged its foot when we were working the Tanami, and we literally pulled the trap apart to find out what went wrong. And it wasn't the trap, it was a peg. The peg worked its way out because we're working in really hard ground, and the animal damaged its leg on that. And so we told the ethics committee about it, and they said, oh, great, you actually worked out what caused it. That makes our job easier. We can see that it wasn't gross negligence or cruelty. It was a freak accident. Make sure that doesn't happen again, continue.
Thank you.
As an invertebrate ecologist, it might seem weird, but I lectured on animal ethics for about 8 years, and the most interesting thing for me was looking at the history of the animal rights movement and animal welfare generally. Cool fact before I actually talk about something serious. Apparently, can you hear me? Cool. Apparently in Ireland in 1643, a law was passed, which was like the first animal ethics law to prevent farmers pulling the wool off their sheep's back because they didn't have a method for shearing and they just ripped it off. And so they had a law that they thought that this caused the sheep distress, and so they stopped it. So from there, it's only gone from from that to Peter to Harry Harlow to what we have today. And if I may bring up the 3 R's. So you can always refine your methods, you reduce the animals you use, and you reuse the animal for different data rights. So you already know that. So you kind of know how to get through animal ethics committees already. Nice work. All right, question. Yeah, over on the side there.
Yeah, I've got a question for Talissa. Are you mainly focusing on one disease present in sheep at the moment? And if you're not, are you hoping to be able to mass roll out technologies for farmers in general to use in everyday practices, or?
Yeah, definitely. There's a number of applications that the body temperature measurements can use. So there's a number of illnesses associated with body temperature increases, same as us, fever and bacterial infections, viral infections, all associated with the change in body temperature, and also secondary infections. So, if you have a worm infestation that then opens the door to other infections, which have the knock-on effect of a difficulty regulating body temperature or a fever response. So this body temperature work has actually got a lot of different applications, and the infrared work that I was doing was reasonably limited to surface temperature associated with fly strike, because that's of a huge interest to the industry to try and detect that at an early stage. But there's a lot of work going on with infrared for looking at lameness and various sort of localised inflammation that can also be picked up, and obviously the wool's just a bit of an issue that we have to try and tackle. But there's definitely hundreds of applications for body temperature alone as part of the work. So it's very interesting stuff. So thanks.
With your study in Sri Lanka, I was wondering how do the chemicals actually react with the kidney?
Oh, I'm sorry, I don't know about these things because this is a kind of medical study, but I'm thinking about the contamination of soils with cadmium and arsenic and how to go for the solution for this problem. Actually, sorry. Yeah.
Also, I've got a couple of questions. Catherine, is there a way, do they burn the crops once they've harvested what they need? And if so, wouldn't that bring some carbon into the soil?
At the research station we're at, generally they sort of just slash and leave everything on the ground to decompose and trip over the next time when you go into the field. Burning, I don't think that's as common practice, Yui. No. So she's the postdoc that works on the project with me, so it's like, yes, there's a senior person here. So burning is not so much done these days, but generally, letting some of the stubble remain in the ground for the carbon to be reintroduced and broken down is what happens there. I can't remember your name. Plants, with the plants and the bacteria.
Yeah, Dane.
I was wondering, do the different plants have different chemicals?
Yeah, so there's a really large range of them. They're usually, you can group them by classes, and for example, the compound that I had up on the end there that I was talking about was, it was from an aeromophila that I was mentioning before, so the emu bushes, and we find those in a lot of different species from that same genus. They're slightly different. Sometimes you see the same thing across the board, and in fact, this is actually a really big problem that I find something antimicrobial, I get really excited, and it's in, it's something like what's called terpene and 4-ol, so basically your tea tree oil, So we already know tea tree oil does its thing. So, yeah, there's basically a lot of chemicals that are produced in lots of plants, but yeah, finding those unique ones is actually really hard. And so we have to use tools to try and, what we say, de-replicate basically what we already know, and that's actually really hard. So it can take me months and months to get to one compound because it's really hard to clean up compounds from plants, get there, investigate it, get really excited, and then discover it's well and truly known. So it's a problem, but yeah. Yep, so plants do produce different ones too, but they also have a lot that they share in common, yeah.
One more question. What did all of you study initially to be able to get to where you are now? As a student, I really like to know this. In 25 words or less.
Greek and Roman erotic. No, marine ecology from a zoology degree.
Cool. I originally did Indigenous health studies at Wollongong many, many years ago, then public health after that, and then went overseas and did something completely different, and then come back and went, I always loved microbiology, and this kind of came back after this long trip and went, I'm an adult, I don't need a HSC mark to get into this now, and got in and then did that honours, and here I am now.
Yeah, I did a Bachelor of Science Biology a long time ago, and I sort of went on to do technical work for many years, and landed in assistance in research and realised that you could actually do animal welfare research, which was really exciting. So I was very lucky to apply for a scholarship and got it, and the rest is history.
I followed my master and bachelor degree in the field of agriculture. Now I'm doing environmental science. I'm going to relate agriculture with environmental science.
I did a double degree in science and education, as you would know tomorrow, one of my former students. And then I did honours in environmental microbiology, so looking at microbes associated with acid rock drainage. Did teaching for a bit, had a break, and now I'm back doing my PhD. So, yeah.
That was a wonderful finish, and I always hate to wrap up Q&As because I know there's always so many burning questions. But it's 10 o'clock, and we need to do other things, and so you can always grab these guys as they come off. But please give them a really big round of applause for their work tonight. So prizes tonight come from a variety of places. UNESTA, the Student Association, CSIRO, Uni, Costa's, Domino Pizzas and Ecological have all contributed to wonderful prizes tonight. And I'd like to invite Natalie Vanderveer up to the stage. She's representing Ecological tonight, who has sponsored the prize for the rapid fire talks. And Nat will present the iPad to the winner, who is Talissa. Well, congratulations. Just take the path of least resistance. Congratulations.
Thank you. Thank you. Thanks.
Hopefully you can use that in your research. Look, more gadgets. You're welcome. Thank you, Nat, and thank you, Ecological. And that, my friends, brings us to a close. Oh, the pizza coupons are in the trivia prize. Yeah, sorry. Missed out. You just got zonky wrong. That brings it's us to a close this evening, and for Pint of Science to a close for 2017. I would like to thank wholeheartedly again our sponsors, CSIRO Inspiring Australia, New England Northwest Regional Science Hub, Ecological, UNE Science and Tech, and the Wicklow, of which there are representatives from all of those here tonight. And I'd also like to say, and some special thank yous to Caitlin Donnelly, Matthew Elsley, Brendan Carroll for their tireless efforts putting up with stressed Dane and ensuring flawless evenings. Thank you very much for this evening. Thank you very much for this year, and have a wonderful evening.